but we are going to be talking about doing flow cytometry so much this semester. Flow cytometry is like one of those things that immunologists read about, think about, do all the time. And so I need to sort of give you the background on flow cytometry and how it works. The plan for next week is that we're going to do some cytometry once you know how it works. So anyway, I need to like tell you the update of how we need to go through how flow cytometry works. Like I said, the plan for next week is that we will actually prep some samples and do some cytometry, provided that my machine is working by then. I've been working on it a lot. Maybe I should just try to build a radio and then I can go from there. So that's going to be the plan for next week, but we need to know kind of how the technique works because it's going to start showing up soon in lectures and papers and all the time. So we got to know what's up. After I kind of go through this information, I do have a handout for you. The handout has 2 parts. It has some questions, and it's due in 2 weeks in lab. The first part is honestly some basic kind of data analysis using what I'm telling you. I'm showing you data from papers and asking you questions about it, things like that. The second part is asking you some questions about your data from next week's lab. So it's sort of an all-together flow cytometry thing. So there's a part 1 and a part 2 in the handout. I've got printouts of it here that I can give you when I'm done. If you want to sit here, because I am not going to be talking for 3 hours, if you want to sit here and work on those problems and ask questions about at least the part 1 questions after I'm done, totally fine. But the whole packet in general isn't due for 2 weeks because you don't have the data yet for the other part. We're going to be talking about, I'm going to talk about some general antibody techniques and then use that to segue into flow cytometry. So I like this in this part of the class for 2 reasons. One, because we're talking about antibodies in class, and so thinking about techniques that involve antibodies makes sense. But also because when we start actually talking more about B-cell biology in the future, a lot of parts of B-cell biology are things that I need flow cytometry to explain to you fully. So all of these techniques include antibodies. Yay! Here's my antibody. And there are tons of techniques that use antibodies that are out there. And so you can see this table that lists some of the techniques that are out there that use antibodies. There's one that's listed here is precipitation reaction in fluids. I've got precipitation in gels. I've got agglutination. I am now remembering that I used to do this in a slightly different way. So I may draw some stuff on the board. But really, with all of these, we're using the ability of an antibody to specifically bind an antigen. And the difference with a lot of these assays is in terms of the sensitivity. So how much antigen do you need before you get a positive result? And what kind of format is your sample in? So do you have a liquid? Do you have a gel? Do you have a solid? Do you have a cell? Like, that's really how these are varying a lot. Just to give you some examples, you guys have talked about Western blots in Bio 250, where you've taken protein samples, you've separated them on a gel, and then you've used an antibody to find a protein of interest out of all of the proteins that were in the whole cell. And so in that case, I have such pretty slides of this that apparently I didn't feel the need to put in this presentation. I have a tube. It's full of a bunch of different proteins shown here as different little shapes. I can run that protein sample on a gel, and I'm going to get a ton of proteins of all different sizes. And then if I want to find my protein of interest, I can take an antibody, bind it to all these proteins, and detect where the antibody was bound. And I'm specifically looking at this tail region to do my detection. So you guys have seen a Western blot before. Sometimes we can do, so this of course requires you to have a protein sample you can run on a gel. You could also have samples in different formats. So there's one that we have where we have a sample called an ELISA, where we just use a little well of a plate and we put the sample in the well of the plate. And then we again bind our antibodies to it and see, will the antibody bind or not? And so it's conceptually the same kind of thing. If you are need to take a at-home COVID test, SARS-CoV-2 test, it's an antibody assay. It's a, I guess I would call it it's sort of a precipitation reaction, though it's not in the fluid. Where basically you have one area right here. You have a filter paper, and you have these two antibodies attached to the filter paper. And oversimplifying, you have a thing that makes some color if it binds to an antibody on the other end of the filter paper. So imagine like a coffee filter or just a dry piece of paper. You put some stuff on one side, you put some stuff on the other side. Then you swab your nose and put some liquid, and you touch that right here. Now you made the paper wet. And if the paper gets wet, the liquid flows all the way across and all this stuff mixes together. And if there was SARS-CoV-2, now the SARS-CoV-2 would bind here. And it would also bind to the color thing. And you get a color all of a sudden, where there used to not be one on your filter paper. And this one is a control that basically binds to anything. And so you always get a line there to tell you that the test worked. And so basically what you're doing is you're just allowing capillary action to flow some liquid, maybe with SARS-CoV-2, maybe not, down a piece of filter paper. It's again, just an antibody assay doing an antibody interaction. This exact same phenomenon that I'm showing you here is also how a pregnancy test works. You are putting urine that might contain a particular hormone right here. It's now flowing down this thing through capillary action and either binding to an antibody for the hormone or binding to a control antibody. They're fancy. They make it so the lines are perpendicular to each other, so it becomes a plus or a minus. But it's that. And if you think about all of your COVID tests where you're waiting for the two lines, That's all. This is all you're doing. And so all of these things are just ways we use antibodies, and we can use them in so many ways for so many different biochemical assays. Yeah. Mm-hmm. So the antigen is only in the test if it was in your nose, because you're going to swab your nose and put it on there with some saline. And so if you have the virus, then now there is antigen. If you don't have the virus, then there's no antigen. Either there is something that binds a control. Either way, there is an antigen that binds a control. But in terms of the SARS-CoV-2, it's a question of was there SARS-CoV-2 in your nose? No, it's detecting whether the antigen is there to bind to the antibody that is on the paper. Yeah.
I know, like, you can use that for a strep test, but there's also the alternative of swabbing the back. Do they then go and use that? Or?
Usually, if they're doing a swab, they then culture. So, not an antibody test in that case. So many things out there are really taking advantage of antibodies and their specificity. For the technique that we're going to talk about, we are going to be trying to answer kind of a specific question, and we're going to be doing it using antibody specificity. And this is a question that you guys have already thought about in Lab 1. So in Lab 1, remember, you had to tell the difference between all of these cells, right, by eye. And it can be hard to tell the difference between them by eye. And sometimes you get things like a lymphocyte. There's a ton of different kinds of lymphocytes. There's B cells and T cells. There's actually kinds of B cells and T cells. I can't tell you if this is a B cell or a T cell. All I know is it's a lymphocyte. So we have some sort of limitations from this process that we talked about. And that's sort of not the only sort of limitation for this, but just to, okay, one limitation is that if I actually looked at blood, white blood cells would be really rare. So I'm looking for needles in haystacks. If I want to do it, if I want to take some blood and study leukocytes and start to study all sorts of different cells, it's a needle in a haystack. You guys all came up with a percent of what percent of the blood was leukocytes, and you all dramatically overestimated because you all picked up a field of view that had at least one in it, so you all got 1%, and it's way less than 1% in reality. So, we're trying to look for needles in haystacks, which is pretty hard. We also have this problem where some of these cells that you saw here are really heterogeneous. So these charts, both this chart here and this pie chart, show you some of the kinds of lymphocytes that exist. And the point of the story is when you were doing that count in Lab 1, you were like, lymphocyte. And you did not do things like try to figure out which of these many kinds of lymphocytes you had. But there are lots and lots and lots of situations where we need to have that kind of information. So you guys, in your answers to the Lab 1 questions, gave me some suggestions of how you could make your data better, how you could kind of deal with some of the difficulties here. And what kind of things did you come up with of things that could help you deal with the difficulties? Yeah, Jonah. Yeah. Okay, so if we had a way to remove red blood cells, then that can make our life a little easier. Sure. Yep. We could use a larger sample. So what's the deal? So like, what would a larger sample help us with? So you could get a lot more accurate if you weren't doing 100 cells, if instead you tried to do 100,000 cells, right? How would you feel if I asked you to repeat lab 1, but tell me 100,000 cells? You're all making some excellent faces about that. So what's the problem with that scheme of doing 100,000 cells? Yeah.
It's just too tedious. It takes too long.
It's tedious. It would take forever, right? It would be not fun. The other thing is that, I'll come back to that. The other thing that we could do is have some way that we could get more information than this about the cell. If we could somehow get more information than we're getting here, that would either make you more certain, or it might help you like determine, is that lymphocyte a B cell or a T cell? So if we had some way we could get more info, that would be cool. And if we had some way that we can get more cells. So how long did it take you to classify 100 leukocytes in lab 1? Yeah.
It took Jonah and I 15 minutes.
Okay, 15 minutes for 100. Okay. Anybody else? Yeah. 16? Yeah. 25 minutes? No, all good, all good. So you see kind of what we're talking about? This technique that I'm going to introduce you to today, when I was a graduate student, every experiment that I did had a minimum of 100,000 cells, and every experiment took a max of 2 minutes. So I was getting 100,000 cells in 2 minutes. No, that's just to put it in the machine. The prep took a little longer than that. But the machine would actually analyze 100,000 cells in 2 minutes. In fact, you can get it to go higher, to go faster than that. So what I hope you can notice here is exactly how powerful this is and how useful this is in doing this analysis that we're thinking about. And this is the type of question that I was trying to address as a grad student. So I was actually infecting mice with some viruses, and I was measuring the number of CD8 T cells that responded to that virus over time to look at primary and secondary responses. So I have graphs that I published that look approximately like this. So I was measuring that virus-specific T cell, CD8 T cell, and making a graph like this. Okay? I legit have published graphs like that. What that means is I had to have a way that I could tell the difference between a CD8 T cell that bound to HIV and a CD8 T cell that bound to a different antigen, like a flu antigen. And not only did I have to have the ability to do that, even if I did as Jonah said, which I did, and got rid of all the red blood cells, I actually had to be able to pick out that CD8 T cell that responds to HIV from all of the other white blood cells in my mouse blood. And I look at this slide and I think about me when I started teaching, because I actually spent the time to like make the number of cells correctly proportionate on this slide. And now I'm like, I sure would not do that right now, but that's okay. So you can see like needle in a haystack. But yeah, if you look at a lot of immunology papers, this is sort of very common, a very common kind of data that we might have. And so the way that we deal with this is that we know that leukocytes have characteristic proteins on their cell surface. So if I think about all of the types of cells that you guys were looking at under a microscope, I can tell you that they have proteins that we've described, that we know of on their cell surface. I'm going to say examples right now. You don't have to like have these examples memorized for later. As we talk about different cells throughout the semester, we'll say what the ones are for that cell. But it's easier if I give you some examples right now. So for example, a T cell has a T cell receptor. It also has a protein called CD3. And immunologists just know that. And it also has a protein called CD8. If it's a CD8 T cell, if it's a CD4 T cell, well, it still has CD3 because it's a T cell, but it has CD4. If something's a neutrophil, it has a special protein called Ly6G. And so there's like a pattern for all of these cell types of proteins that are on their surface. Over the years, immunologists have spent a bunch of time cataloging the proteins that are on the surface of different leukocytes. And so you might imagine that you all took your favorite leukocyte and found the proteins on its surface, or started finding proteins on its surface. And so let's imagine that, Brenna, what's your favorite leukocyte?
Oh, I don't know. What's your favorite?
That's a really hard question.
Easy one.
Does anyone have a favorite leukocyte? I need at least 2 people with favorite leukocytes for my example. A natural killer cell. All right. Brian really likes a natural killer cell. So let's imagine that Brian and Josh both really like natural killer cells, okay? And they start describing proteins on the surface of the natural killer cell. Brian, you just found a cool protein on the surface of a natural killer cell. What would you like to name it? You can name it whatever you want. Right, the Josh and Brian protein. Now, Josh, you have a separate lab. You have just found a protein on the surface of a natural killer cell. What's the name of your protein? NK16. All right, so we got NK16 and we got the Josh and Brian protein. Here's the question. Is NK16 and the Josh and are those the same protein with 2 different names, or are they one protein? Or are they 2 different proteins? You don't know, right? It sort of becomes a little bit tricky because we don't have a standardized way of doing this process, a standardized way of naming. For a while, we didn't have a standardized way of naming. It led to papers like this. I had to read this paper as a grad student. They had to list every name that the protein had ever been given. So in this case, the CC chemokine, thymus-derived chemotactic agent 4, there's one name. Secondary lymphoid tissue chemokine, there's a second name. 6C-kine, there's a third name. Exodus 2 is a fourth name. So they had to list all the names for this protein because it didn't necessarily have a standardized way of naming it. That seems lame, right? And it also seems like if you were a researcher, so now Jonah also loves NK cells, and Jonah decides he's going to start doing some experiments on NK cells. He reads Brian's paper, he reads Josh's paper. He tries to figure out his experiments, and he's like, I don't know if these are the same proteins or not, and I don't know what to do, and I'm confused. And it's annoying, right? It's all very annoying. So what immunologists did is they came up with a standardized naming system for these proteins. There is actually a group who gets together once a year to look at all the proteins people have found on the surface of lymphocytes and give them standard names and figure out like which ones are all the same and which ones are different. You can tell the people who are part of this committee must be really fun at parties. If that's what they really want to spend their time doing. But whatever. And this is called the CD system. And so the CD was originally named as cluster of differentiation because people thought something might be going on with different differentiation states. No one ever says cluster of differentiation anymore, just go with CD. But basically, we just made a list. So we said, aha, we found a first protein that's on a leukocyte. We're going to call it CD1. And then somebody else found one. So Brian's protein now gets called CD1. And if we figure out that Josh's protein is different than Brian's, we call it CD2. And if we figure out that Jonah's is different, we call it CD3, and so on. We're up to about CD400 for most immunologists right now. The back of most immunology textbooks have a CD chart. In my lab, I have CD charts in my office. Do you need to have the CD charts memorized? No, I'm going to tell you about the important CD molecules that are important for a particular cell type as we get to that cell type. But one of the things people are always, when people make fun of immunology, they're like, oh my God, those immunologists, they love their CD jargon. It's just putting a number on things so things aren't annoying. It's not like trying to be all impenetrable. It's just that we're trying to be a little bit organized. And so we have CD1, CD2, CD blah, 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 blah. So one of the ways that we can address this needle in a haystack problem is we know some of these characteristic proteins on the surface of a lot of these cells. And they have these CD names in most cases. You can see there are some that don't have a CD name listed there, but that's because the old name stuck. They actually do have CD names too. So we can actually look at these proteins on the surface of cells to help us know which cells are which. And you might be like, awesome, that's great, Dr. Barker. How do you look at a cell and know that it has a protein called CD8 on its surface? Those glasses you're wearing must be extra special if you can actually see CD8 proteins on the surface of a cell. And the way that we actually really do this, maybe I should push the right thing, is use antibodies. So we have antibodies against all the CD markers. And so we'll take our cells of interest and we will add antibodies to them. And usually those antibodies have a modification where they're going to be fluorescent. So for example, if I wanted to detect a CD8 positive T cell, I know that T cells all have CD3 on them. CD8 T cells have CD8. And so I could use an antibody against CD3 that is blue. It has a blue marker on it. And that's going to find all the T cells. Going to turn all the T cells blue. And I could use an antibody against CD8. That's going to turn all the ones with CD8 red. And so I can look and see, well, how many cells are both blue and red? And those are going to be my CD8 T cells, is basically how this is going to work. Does that part make sense? Do we get what's going on here? Yeah? Okay. So that's all cool. This is a big part of what we're doing, but there's still one other piece to this. Because you might still look at this and be like, awesome, Dr. Barker. Now we're going to look at these under a microscope, and we're going to, instead of looking for the things we were looking for before, look for blues and reds. Do you think that that is going to speed you up very much compared to what you did before? No. So it's a way you can get more information about the cells. Yay. But it's not going to help you with your speed problem. If really all you're doing is, hey, let's look at cells under a microscope and see which color they are. That's not helping you with the speed at all. It's going to be just as long and just as annoying, even though, yes, you're getting more information. So the other useful part of this is that we have a machine that speeds this whole business up. And that machine is called a flow cytometer. And so here is a little bit of info about the flow cytometer. I'm going to explain it, and then I'm going to show you a little video that talks through it. So what we do is we first take our cells and do some pipetting and stuff with them. We prepare the cells. So up here you've got, your sample, which is your cells that you've prepared. What you might have done in preparing those cells is you might have added antibodies of interest. So you took the spleen cells from your mouse and you said, I want to know about CD8 T cells in my mouse, so I'm going to add my anti-CD3 and my anti-CD8 antibodies. So I'm going to mix it all together. And you could see that if I did that, some cells would have no antibodies bound to them. Some cells might have one. Some cells might have multiple, all depending on different proteins that are on the surface of those cells. And I'm going to mix my sample with a huge excess of liquid. Notice that my sample up here is a heterogeneous sample. So it's not like I purified the sample at the beginning. It's just the whole mixture of all the cells that were in the blood or the spleen or the whatever mixed together. So again, I basically just threw a whole haystack in the machine, and I'm hoping that the machine is going to tell me where the needle is. And what the machine is actually able to do is use changes in pressure and other electrodynamics to make the cells fall in droplets of one cell per droplet. And so the machine is going to take this whole mixture. I'm going to type on my computer a little bit, and I'm eventually going to get the machine to drop the cells. You can see the sort of cells dropping right here. One cell per drop are just going to get dropped through this area. So here you can see the stream of fluid drops containing cells. Some drops might contain this cell that had no antibodies on it. Some drops might contain a cell that had one antibody on it. Some drops might contain a cell that had multiple antibodies on it. And this stream of droplets is then going to be hit by a laser. So it's basically going to go into a laser. And the way that the cell and any antibodies attached to it interact with the laser light will be detected on a computer at the end. And so I'm going to then take the information that comes from that computer, and I'm going to use it to give me information about the cells. And again, this is where I can set it up so the speed of those droplets is letting me get 100,000 in 2 minutes, because it's going to go really fast and just shoot them with the laser. The computer is going to read out information about how that cell interacted with the laser light. We're going to move forward. So that's the sort of broad overview of cytometry. Let's see if this actually works.
Flow cytometry is a powerful technique for the analysis of multiple parameters of individual cells within heterogeneous populations. Flow cytometers are used in a range of applications, from immunophenotyping to plethysmography analysis to cell counting and GFP expression analysis. The flow cytometer performs this analysis by passing thousands of cells per second through a laser beam and capturing the light that emerges from each cell as it passes through. The data gathered can be analyzed statistically by flow cytometry software to report cellular characteristics such as size, complexity, phenotype, and health. In this tutorial, we will look at how a flow cytometer works, how scattered light and fluorescence are detected by a flow cytometer, and how the resulting data can be analyzed. This view shows the primary systems of the flow cytometer schematically. These are the fluidic system, which presents samples to the interrogation point and takes away the waste. The lasers, which are the light source for scatter and fluorescence. The optics, which gather and direct the light. The detectors, which receive the light. And the electronics and the peripheral computer system, which convert the signals from the detectors into digital data and perform the necessary analyses. The interrogation point is the heart of the system. This is where the laser and the sample intersect, and the optics collect the resulting scatter and fluorescence. First, let's talk about how the sample is delivered to the laser. Here we see how the sample is transported through the interrogation point. For accurate data collection, it is important that particles or cells are passed through the laser beam one at a time. Most flow cytometers accomplish this by injecting the sample stream containing the cells into a flowing stream of sheet fluid or saline solution. As you can see, the sample stream becomes compressed to roughly 1 cell in diameter. This is called hydrodynamic focusing. In fact, flow cytometers can accommodate cells that span roughly 3 orders of magnitude in size. In most cases, cytometers will be detecting cells between 1 and 15 microns in diameter, although through the use of specialized systems, it is possible to detect particles outside this range.
So what I hope that you sort of got from there is this idea of how we're going to put this heterogeneous sample in our machine, and how we're then going to focus it 1 cell per droplet through our stream that's going to go through the laser, and that we're going to get some additional information about. Now I'm going to try one more thing. Let's hope it works.
Now let's see how laser light is used to detect individual cells in the stream. As a cell passes through the laser, it will refract or scatter light at all angles. Forward scatter, or low-angle light scatter, is the amount of light that's scattered in the forward direction.
I'm going to draw this on the board in a second too.
The magnitude of forward scatter is roughly proportional to the size of the cell, and this data can be used to quantify that parameter. But how can we record this scattered light? Light is quantified by a detector that converts intensity into voltage. In most cytometers, a blocking bar, called an obscuration bar,
no one cares about that,
is placed in front of the forward detector.
I want to know about
The obscuration bar prevents any of the intense laser light from reaching the detector. As a cell crosses the laser, light is scattered around the obscuration bar and is collected by the detector. Because small cells produce a small amount of forward scatter and large cells produce a large amount of forward scatter, the magnitude of the voltage pulse recorded for each cell is proportional to the cell size. If we plot a histogram of these data, smaller cells appear toward the left and larger cells appear toward the right. A histogram of forward scatter data is a graphical representation of the size distribution within the population, but such a graph only presents one-dimensional data.
So here's a bunch of tiny cells and here's a bunch of big cells.
Side scatter. As we have already seen, a cell traveling through the laser beam will scatter light at all angles.
So what this just told you about is one of the ways that the light interacts with the cell. So when I am using my flow cytometer, I am able to look at the way that the light interacts with the cell in 2 ways. Basically, those 2 things are telling me 2 things about the cell itself and its physical parameters. So they don't require any antibodies, they don't require anything. It's just that different kinds of cells interact with the laser light in different ways. And you just heard about one of the 2 parameters, the 2 physical parameters that we look at. That physical parameter is called forward scatter. And the way I like to think about forward scatter is it's basically how much the light is bent around the cell. So you can see I might have a small cell. It has a small forward scatter. If I have a big cell, it has big forward scatter. And I can plot, if I have a graph and I put forward scatter on it, my small cell was here, my large cell was over here. And I put them both low because there was only one of each. Though there are different ways I could do that graph. We'll get to the graphs in a second. So this is sort of what we're getting with forward scatter, and that's what the video just explained. There is a second physical parameter that we also like to look at. And so when I think about forward scatter, I'm thinking about size. The other parameter that I often will look at is, as the slide kind of already tells you, side scatter. And the difference with side scatter is it's not measuring the size of the cell, but it's measuring how much stuff is inside the cell. Sometimes people call side scatter a measure of internal complexity or I was always taught it as granularity. This is one where I definitely need the video to give me like a real drawing. I'm going to show you some things, and it's, those of you who know some about physics are going to be like, yeah, that doesn't make any sense at all. But that's because I can't draw. But basically, the idea is some cells have very few granules, don't have much stuff inside them. And some cells have a lot of stuff inside them, like a lot of granules. Remember our granulocytes? And if we shine light on this cell with nothing inside it, it's not really going to get bent through that cell. If we shine light on this cell that has lots of things inside it, it's going to bounce around off of all this stuff. And that light's going to be different. And so basically we're looking at how much does the light bounce around because of stuff inside of the cell, or how granular is the inside of that cell. And so we can distinguish every cell that goes through our flow cytometer based on its size. Is it big or small or medium? And based on its granularity. Does it have a lot inside or a little inside? And so this will now say about side scatter. If I can hit the play button.
Light scattering at larger angles, for example to the side, is caused by granularity and structural complexity inside the cell. This side-scattered light is focused through a lens system and is collected by a separate detector, usually located 90 degrees from the laser's path. The signals collected by the side-scatter detector can be plotted on one-dimensional histograms, like we saw for forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example,
so if this was a side scatter plot, these cells have low side scatter. They're like between 0 and 50. So they probably don't have very much stuff inside them. These cells are kind of medium. They're like between 50 and 100. So they have some, and these cells are really high. And so they have a lot of stuff inside them. This is just showing it as a histogram, so how many cells are in each group. And I could have paused the other thing about, and it was doing the same thing of how many were big, how many were small. What is also really cool about this is we don't just have to look at the data in a histogram. I could change this. Instead of having cell count on one axis and a parameter like side scatter on one axis, I can have two different parameters on the two axes. So I could have forward scatter on one axis, side scatter on the other axis. And that, if I did that, I'd get a plot that looks like this. I went to grad school on the East Coast, so I draw my plot this way. I have since learned that people who will go to grad school in California draw their plot differently, but that's fine. This is how the plot works, because I say so. And so I'm gonna, we'll say I might look at 100,000 cells. And the first cell might be kind of tiny and not have any granules. And so it would be low forward scatter and low side scatter. And then I might have another cell that was huge and had tons of granules. So it could be high forward scatter, high side scatter. And then maybe I'd have another cell that was tiny with lots of granules. So it might be up here. And I'd have another cell, and I could actually go through and I would have a dot for every one of my 100,000 cells telling me both how big and how granular that cell was.
Scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram.
Just some small guys.
Multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots.
No, stop,
now let's see how laser light is used to detect individual cells in.
So here, this is their side scatter. They've got the not so granular, the medium granular, the very granular. This is their forward, they had forward scatter where it was kind of like mostly smallish, a few really biggish. And here they've put the same things on, this on two graphs, and so, or on two axes. So they have really small stuff. They've got some, small stuff that's not super granular. They've got some bigger stuff that's more granular. And it's only a little bit bigger. You can look at things in a lot of different ways like this. And each one of the dots on that plot is a single cell that we're able to get data on. Whoa. This video is going to make you mad again.
Now let's see how laser light is used to detect individual cells in the stream, forward scatter. The one-dimensional histograms we have seen so far do not necessarily show the complexity of the cell populations. For example, what appears to be a single population in the forward scatter histogram is, in reality, multiple populations that can only be discerned by looking at the data in a second dimension. This is done through the use of two-dimensional dot or scatter plots. You can see that the peaks from the forward and side scatter histograms correlate with the colored dots in the scatter plot. Now we can view the results obtained when we create a scatter plot using forward and side scatter data from a typical peripheral blood cell run. The populations that emerge include lymphocytes, which are small cells possessing low internal complexity, monocytes, which are medium-sized cells with slightly more internal complexity, and neutrophils and other granulocytes, which are large cells that have a lot of internal complexity. This multi-parametric analysis is the real power of flow cytometry. Now let's take a look at another parameter that can...
No, we're not looking at that parameter right now. So this is actually some data that I generated as a grad student. So this is blood from an animal where I just ran it, and you can see all of the dots. I'd have to look back at this to tell you exactly how many cells I ran that day. We'll say it's a couple 100,000. And so you can see all of them. You can also see that there are some places where there's dots are kind of more denser than others that we often think about as populations of cells. Yeah, there's like a weird cell that's over here, but there's also kind of places where there's big populations of cells. And sometimes it's actually a little hard to look at. You're like, okay, is this area denser than this area or not? I don't know. At some point, it's just black. There are other ways you can actually make it look pretty, where you can have contour plots of actually seeing how dense the dots are in different places, or you can actually color code it based on density, where you can get some more information about those plots. I know that there's definitely one paper that we're going to talk about this semester where they use contours. I actually find contours to be very old-fashioned, but it's an old paper, so. I like to use ones where you can actually see the individual dots, but you also get the colors to tell you how dense it is. And so if you look at this, what you can notice is it looks like there's 3 groups in these cells. Yeah, there are some weird outliers, and I'm not going to try to figure out what those are unless that was my experiment, but there are just these 3 main groups. And I will, I can actually even use my software and draw a little gate around each of them, draw a little region. And the software will even say 60.7% of the cells were in this region you drew. So 60.7 of the cells have this kind of phenotype. 3.81% of the cells have this phenotype, 28.4 have this phenotype, and I can draw that. So that starts to give me pretty good information. I could compare different samples. This population of cells that is sort of the smallest but totally the least granular, those are lymphocytes. So I know that this animal had 60.7% of its blood, its white blood cells were lymphocytes. 3.81% were monocytes, because this is a known monocyte population as sort of the largest ones that are sort of medium granular. This very granular, kind of medium-ish size, are the granulocytes, largely neutrophils, because we're looking at blood here. And so I can get that data in a minute. by just throwing my cells. I didn't even have to put antibodies on them. But because I did put antibodies on them, I can get some additional information. Because the laser light is going to interact with the dye that I put on the antibody and is going to excite it to emit light at a known wavelength. So my laser light is basically going to hit this thing, the thing is officially called the fluorophore, and make it emit light. And so not only can I capture forward scatter and side scatter, I can capture all the light that comes off of any of the fluorescence molecules that I have attached to my cell. This is the energy state diagram that they were going to tell you. I guess, how long is it?
One of the most common ways to study cellular characteristics using flow cytometry involves the use of fluorescent molecules, such as fluorophore-labeled antibodies. In these experiments, the labeled antibody is added to the cell sample. The antibody then binds to a specific molecule on the cell surface or inside the cell. Finally, when laser light of the right wavelength strikes the fluorophore, a fluorescent signal is emitted and detected by the flow cytometer. How is this fluorescence information collected? The fluorescent light, coming from labeled cells as they pass through the laser, travels along the same path as the side scatter signal. As the light travels along this path, it is directed through a series of filters and mirrors so that particular wavelength ranges are delivered to the appropriate detectors. Fluorescence data is collected in generally the same way as forward and side scatter data. In a population of labeled cells, some will be brighter than others. The voltage pulses are recorded and can be presented graphically.
And so in this case,
a two-color experiment.
This case, these cells only have a little bit of my protein of interest, and these cells have a lot of my protein of interest, based on just sort of the histogram of amount of fluorescence of my protein of interest. How bright was it? How much did we excite that color? And so we'll for example, talk about, we're going to put in an antibody with a green thing on it. These cells only had a little bit of green. The antibody only bound them, so that target protein was only on them a little bit, or was not on them. These cells are very green, so they have my target protein of interest.
What if we want to do a two-color experiment? If we analyze data from the two-color experiment using a scatter plot, 4 distinct populations emerge. Looking at the dot plot in terms of quadrants, cells with only bright orange fluorescence appear in the upper left quadrant. Cells with only green fluorescence appear in the lower right quadrant. Cells with both bright green and bright orange fluorescence appear in the upper right quadrant. And finally, cells with both low green and low orange fluorescence appear in the lower left quadrant. Multiple fluorescence parameters are necessary to dissect complex biological systems.
Okay, so you saw this example where we're going to have these different colored things. The example you just saw was green and was orange, but it could actually be whatever colors my machine can read. And so when you buy your machine, when you buy your machine, you might say, okay, I want a machine that can read green, yellow, this orangey color, and this pinky color. Yes, the dyes have names, which are labeled here. You don't care about the names of the dyes. And so then I could actually look at, is my cell green and orange? Is my cell green and yellow? Is my cell green only? Is my cell none of the 4? And I can start to get all of this, whether it's green or orange or whatever, kind of depends on the machine I have. And so if I used a machine that was sort of like this, I would get for every cell, for every one of my 100,000 cells, I would get 6 measurements off that cell. I would get a forward scatter measurement. I would get a side scatter measurement. I would get a how much green. I would get a how much yellow. I would get a how much orange. And I would get a how much red. And so I could get all of that data on all of my cells within a couple minutes. And so hopefully you can see that that's super powerful. The cytometer that I have does not do 4 colors. It's slightly less than 4 colors. The most complicated cytometers that I've ever used was a 19 color. I have generally said that the most complicated one that exists is 50. But that's honestly not true, and I know there are some even more crazy complicated ones, but I'm not going to deal with, because they start having other things added onto them and all sorts of bells and whistles. So, like this 4 color business is pretty often what we see. And so basically we're getting forward scatter, we're getting side scatter, and we're getting whatever colors for each cell, and then that's going into our computer to give us data on all of those cells. What I also want you to notice is that after the cells go through this analysis, they end up in the trash can. So you can see my very well drawn trash can here at the bottom. I will come back to that in a second. And so we can look at the data, as you've seen, in a couple different ways. We can look at it with individual dots, where we are actually picking any 2 parameters. So here I basically picked orange and green and looked at my cells and how much orange and green they had. And I said, oh my gosh, I've got 4 different groups based on high or low amounts of orange and green. I can also just take them and be like, okay, I just want to know orange. Are they high or low on orange? And I can look at a cell count histogram. Or how high or low are they on green? And do a cell count histogram. So they showed you in the video how this can sort of like go side. They showed you histograms sort of going sideways and matching up with the dot plot. And I can't do that. But if you need to see it again, these slides are posted. And you can watch the video. And the other really cool thing that you can do is, as I showed you, you can actually draw little regions with your software. And the software will tell you things like, oh, this % of the cells are in the region that you just drew. So you said you care about cells with a forward scatter of 200 to 600 and a side scatter of less than this. You care about cells in this circle. Oh, that's 60.7% of all the cells that were in the blood. So that's cool. Some interesting info. But then you can do something else. Officially, these little regions or these little areas or groups of cells that I've drawn are called gates. I could, if I looked at this, say, oh, cool. So I know those are lymphocytes. I know those are monocytes. I know those are granulocytes. I know that because I've done this a billion times. I actually today only care about lymphocytes. I don't care about the other ones. I only care about lymphocytes. You know what I would do in that case? I would double click on those lymphocytes. And I could then take just the cells that were there and look at them on 2 other parameters of the many parameters I was looking at. And I could say, well, of just the lymphocytes, now that I've gated on the lymphocytes, so I am looking at only the lymphocytes, I can further subdivide the cells that were in this group. And so this is actually a situation where I did that. So here I did further analysis of the lymphocytes. Here I did further analysis of the monocytes. Here I did further analysis of the granulocytes, looking for the expression of 2 other proteins on them. And you can see that by and large, the granulocytes didn't have those other proteins. They were kind of low for this one and low for that one. But there were definitely 4 nice, robust populations in my lymphocytes. And I could then say, oh my gosh, I only really like these lymphocytes. Let me double click on that and examine them with further stuff. If I have a machine that does 16 colors, I can like super finely distinguish all of these things. And I could look at these guys and then these guys and then these guys. I could do like days of analysis of looking at different types of cells. And so you can see this here as well. So this might be, you might say, oh, I really like this population of cells here, this 24%. I'm going to get rid of the other ones and only analyze that 24% to see how much of these 2 proteins, CD8 and CD3, they have on their surface. I only really care about the ones that have both, so now I'm going to look at them further. Or I only care about these ones that have CD4 and CD3, so I'm going to look at them further. And so we can use our machine to actually subdivide that 100,000 cells and start to find out about amounts of different cells. You can see that each time I'm getting a percent, which is basically what percent of the cells that were in this whole plot are in this gate that I've drawn. So I can actually end up getting a ton of information about this. This is what some actual flow cytometry data looks like. And this is the another view of the same thing. These are both very classic. This is a classic plot that that a lot of immunologists know well. When I was a second year grad student, we always had this event, and the second years had to design a T-shirt for everyone in the immunology program. And every year, the second years would design a T-shirt, and the person who was in charge of the program would throw it in the trash and design it himself because he hated whatever we drew. So my class was like, we're not putting time into this. And so that's what we gave him. We just gave him this picture, and he thought it was the coolest thing he'd ever seen in his entire life. And so we all have pictures so I now have a T-shirt with this randomly on it. It's something that you see when you look at the thymus, and so our class called it the thymus bird and said it was our mascot. So that's the thymus bird. And I also want to show you, a couple things that you can see here. Oftentimes when we look at plots like this, though it doesn't have to be, we tend to draw our gates like this, where we make quadrants. So we often will do it like this, though it doesn't actually have to be. We can see, we get percentages of how many cells are in each quadrant. Let's imagine, all right, so this one we'll say is 80%. This one's 12%. This one's 3%. Let's imagine this one wasn't labeled, because sometimes in some papers, they don't label all of them. Usually the one they don't label is that one, but let's imagine they didn't label this one. Is there a way we could figure out what percent of the cells were in this quadrant from the other information that we have? I see lots of head shakes. How would we figure that out? Yeah, Brenna. Yeah, if we have quadrants, then we know they should all add up to 100. So we could just take 100 and subtract each of these and find out how much is left. If I wanted to know, well, how many of these cells have any CD8s on them at all, I might add up 80% plus 5% and say it's 85%. If I wasn't out of 100, then I'd have to take 80 plus 5 divided by all of them. If I wanted to say how many of the cells that have CD8 also have CD4, I could do 80 divided by 85. So I can get a lot of information from something like this. And so you can see that basically when we do a quadrant gate, the bottom gate, or the bottom left, a lot of times this semester we're going to see quadrant gates. The bottom left is minus minus. It's minus for both things. Sometimes we might say low instead of minus, but low or minus, either way is fine. So you can see that. So in this case, 3% were minus minus for the two proteins. Here we have plus for one of them, minus for the other one, plus for CD8, which we can see from the axis, minus for CD4. It's about 5%, 12% that are CD4 positive, CD8 negative, and 80% that are double positive for CD4 and CD8. So I don't care that you know those numbers. I care that you know how to read them off of this graph. If I were going to draw this on the board, you guys are well aware of my awesome art skills. And so I also want to show you how I would draw this exact same plot. And one of the reasons I note this is because I'm going to be drawing them a lot. I also will ask you from time to time to draw them. And so I want you to know the things that are sort of key in a drawing of one of these plots. So if I draw that exact same plot, I need to draw axes and label the axes. So I get some partial credit just for that. And I'm not going to necessarily tell you you should have the quadrants or you shouldn't. I'll make them right now. I usually don't care if you actually have the quadrant lines or not. But what you can see in this plot is that this population of cells, the population of cells in this quadrant, is pretty rare. It's the smallest percent at 3%. So I'm going to draw that as a small circle. This one's 5%. It's a bit bigger. So I'm going to draw that as a circle that's a little bit bigger. This one's 12, so that's even a little bit bigger. And this one's 80, so it's going to be really big. And so I'm usually going to be caring a lot that you have axes that are labeled and that you have circles that and have appropriate sizes. I am not going to like have my millimeter ruler out and care about them. If I was looking at this, I would really care that this one was the tiniest and this one was really big and these were sort of like in betweenish. But I'm not going to like really get too deeply into it. So that's what all I would want for a drawing of that plot. Sometimes if a type of cell is not present in a particular situation, there should be nothing in that quadrant. So sometimes a quadrant could be blank. That's okay. As I mentioned, I have like one more thing I want to tell you about flow cytometry, and then I'm just going to give you the problems. And you can work through all of the part 1 problems. You know what you need to know. I'm happy to help you with them as much as you want. The part 2 is going to be based on the data that you generate next week when you do cytometry. So just so you know, this is actually what I did as a grad student, is I took blood samples from mice every day after different infections. And then I first got the lymphocytes and then looked at how many of the lymphocytes had CD8 and how many of them could bind to my antigen of interest. They had a specific kind of receptor on them. And so I would just run cytometry on the blood every day, get a percent, and then put that on my graph. And I can get all sorts of fun graphs really easily this way. So there's one more slide. And the one more slide is interesting to me in 2 ways. One, it is a modification of flow cytometry that many people use. And in fact, people are using it more and more and more and more. And even like not immunologists are using it all the time now. So it's cool. It's really useful. It's also fun because it allows me to tell you a fun fact about all of this. And I really enjoy this fun fact. So I am always going to talk about all of this as flow cytometry. Some people talk about everything that you've seen here as facts, shown here listed as facts. If you hear about facts from people who are not being super picky like me, they might mean flow cytometry, what I've told you guys as flow cytometry. If we're being super technical, facts is a specialized kind of flow cytometry. And it's a specialized kind of flow cytometry called fluorescence activated cell sorting. So the idea with fluorescence activated cell sorting is that you do all the things that we've been talking about with flow cytometry. But there's an additional thing that happens at the end. Remember before, the sample went into the trash can. Now, the sample's not going into the trash can. Now, here's our droplets with 1 cell per droplet that are coming out of the machine. And when they come out of the machine, they're actually going to get an electrical charge added to them. So they're going to be positive or minus. And the computer is going to figure out some information about them. And I am going to tell the computer, I want cells that have these proteins on their surface. I would like cells that have just protein A and protein B on its surface. Or I would like cells that have other things. The computer is going to figure out what that cell is while the droplet is falling, and then it's going to take these two plates here and switch around the electrical charges to make the droplet fall in a different direction. And so, in fact, the cells can be sorted based on the proteins that they have. And I can get collections of like cells with only one color, or cells with two colors, or cells with 0 colors from sorting with my machine. This addition onto a machine is makes it way crazy expensive. But you can get sorting, and then you can take these cells and go do other experiments with them. And so there are lots of situations where people will sort out particular types of cells from a homogeneous mixture, or from a heterogeneous mixture, using fluorescence activated cell sorting. Do you have a question?
I was gonna say, is there issues then that comes with that?
What kind of issues?
Like, are there things that have to be able to use them after? Would there be things that would affect them, like, I don't want to say like contamination?
So yeah, you do. So you usually do it under some special aseptic conditions to deal with the contamination, and you do have to think a lot about your experiment and what you're doing afterwards and how you want to set it up. So there's a lot of planning that goes into all of that, is what I will say on that. So, but I still haven't told you I'm going to try one thing. It's probably going to fail. And then after that, I'm going to tell you my fun fact, which is that once upon a time, somebody made a really cool video. But I think if I remember correctly, this link is dead. But we're going to just try it just in case. The link is dead. Okay. Yeah. That's fine. You can imagine that you don't need to see the video. But I can still tell you my fun fact. So these machines were invented in the late 70s, early 80s. And at the same time that this machine was being invented, there were some other machines that were being invented for not immunologists. There's this one other machine that was invented for not immunologists, for people who worked in random offices, where like, Brenna could have a piece of paper that she would like to send to someone in California through the phone. So she could take her piece of paper and she could put it in the machine and she could have it get sent to someone else, like in California, through the phone. And it would be a copy of that piece of paper. So what's another word for copy? Some of you might see where I'm going, so yeah. So how about, I'm going there. Okay. Yeah? We're getting there. You guys are right on the right track. So it's because the word is honestly so old-fashioned, you probably haven't even thought of it. So there's a word for copy that exists. Called the facsimile. Yeah, so it's a word that means copy. And so these people made a machine so they could send people copies of the paper through the phone line. And they were so excited they could send people a facsimile. They were so excited. And let's imagine you invented a machine called a facsimile machine. And that is actually the technical name of the machine, is a facsimile machine. What would you call that machine that when you try to get a patent for it? A fax. How would you spell fax? F-A-C-S, right? Because that's the beginning of facsimile. Guess what? When they tried to get that patent, the patent was already for FACS was already taken because this was invented the year before. And so this took the patent for FACS fax. And so the company who was doing all of this was Xerox, and they were like, crap, we can't use FACS. What are we going to use instead? Well, there's a bunch of X's in our company's name, so let's name it fax with an X instead, because our name that we want to use got taken. So that's where the fax machine came from, and why it's called fax, and why it has an X in it instead of having a CS, because we stole it, because we're awesomer. I know, you guys are like, yes, she is the nerd who was on Jeopardy. So those are my random facts about that. And I'm just going to give you guys this worksheet. If you want to work on it now, you should. If you don't have to, but I'm happy to help you if you're working on it now. It's due in 2 weeks.